Dinitrogen is the major constituent of the Earth's atmosphere, representing the largest global pool of nitrogen. While nitrogen is essential to all life, but the vast reservoir of dinitrogen in the atmosphere is unusable by most organisms. Basically, nitrogen fixation is a chemical process by which molecular nitrogen consisting of strong triple covalent bond in the air is converted to ammonia or related nitrogenous compound. There are different ways of nitrogen fixation. First, atmospheric nitrogen fixation, in which lightning produces enough energy and heat to break the strong covalent bond between nitrogen molecule and allowing the nitrogen to react with oxygen, forming nitrogen oxides. The second way of nitrogen fixation is industrial Haber-Bosch process. It requires high temperature about 450 degrees Celsius and high pressure greater than 200 atmosphere. Hence, this process is extremely energy demanding and utilizes approximately 1% of the total fossil fuel used globally. Now, the other major pathway of reduction of dinitrogen is through the action of selective microorganisms called diazotrophs that carry out a process called biological nitrogen fixation. Only certain species of bacteria and archaea can fix nitrogen, and a list of some important nitrogen fixing organisms is given here. There are two groups of nitrogen fixing bacteria. First one is free living nitrogen fixing bacteria, which carry out 30% nitrogen fixation. This group includes obligate anaerobes, example Clostridium species, facultative anaerobes like Klebsiella species, obligate aerobes such as Azotobacter species, photosynthetic bacteria which includes Rhodobacter species and many cyanobacteria which includes Nostoc species, and some of the methanogens are also included. While the second group is symbiotic relationship bacteria. This include Rhizobium associated with leguminous plants, Azospirillum species associated with cereal grasses. Now, the nitrogen fixation is catalyzed by an enzyme complex called nitrogenase. This enzyme is composed of two components. Component 1 is dinitrogenase, which is a tetramer of alpha 2 beta 2 subunit molybdenum iron protein complex, which is encoded by NIF-K and NIF-D genes, while the component 2 is also known as dinitrogenase reductase, which is a gamma 2 subunit which consists of iron protein which is encoded by NIF-H gene. Nitrogenase enzyme is oxygen sensitive, which means it gets blocked in the presence of oxygen. So, there is need to block the oxygen, which is done by leg hemoglobin protein which is present in the root nodules. These two component protein work together to catalyze the reduction of dinitrogen in a complex reaction with an ideal reaction shown as follows. Nitrogen plus 8 electron plus 16 ATP plus 8 hydrogen gives to 2 molecules of ammonia plus hydrogen plus 16 ADP with 16 inorganic phosphate. This nitrogenase enzyme is well studied in Klebsiella species. Now, here you can see the arrangement of different protein component. This NIF-K and NIF-D complex encodes for the component 1 as we have seen earlier, and this NIF-H is the smaller component of component 2, while the NIF-J and NIF-F are the accessory proteins. Now, the NIF-J is the first protein which convert the pyruvate plus CoA to acetyl CoA plus carbon dioxide is released. Then in NIF-H region, the ATP is hydrolyzed to ADP and inorganic phosphate, thus generating the energy for the component 1 to convert nitrogen to ammonia. So this is the whole process in which the nitrogen enzyme works. Now, here is the location of nitrogen fixation genes, that is NIF genes. We can see here that NIF-J is at first position. Then there comes NIF-H, NIF-D and NIF-K is present. But the NIF-F is situated downstream of NIF-J. But still you can see the NIF-J and NIF-F directionality is the same, and NIF-H, NIF-D and NIF-K have similar directionality, which is important in this case. Symbiotic nitrogen fixation process. Bacteria live in the roots of the legume family plant and provide ammonia to the plants in exchange for the plant's carbon and protected home. Root nodules are the organs induced on most species of legume plants by symbiotic nitrogen fixing bacteria, that is rhizobia. Now, the different steps in root nodule formation is represented in this diagram. So here you can see overall morphology of root, which consists of root hair. There is presence of a small population of rhizobia, which is a group of nitrogen fixing bacteria. So initially there is a small population of rhizobia near the root hair. So what happened during the infection, this rhizobia is supposed to divide and increase its population near the root hair. When the population of this rhizobia has been increased, at that time the root hair will start forming a curl due to the high amount of rhizobia near the root. Now, the rhizobia will infect the root hair and make its entry into the cell of a root. The next step is the formation of infection thread. So, this infection thread is going to be filled with proliferating rhizobia, which elongates through the root and the cortical cell layer. When the infection thread reaches the specialized cells, mainly in the cortex, it stiff fuses with the plasma membrane of the host cell, releasing the bacterial cell that are packed in a membrane derived from the host cell plasma membrane. The branching of an infection thread inside a cortex cell or the root cell enables the bacteria to infect many cells. So at first, the bacteria continue to divide and the surrounding membrane increase its surface area to accommodate this growth by fusing with a small vesicle that you can see in this figure. Soon after this, Upon an undetermined signal from a particular plant, the bacteria stop dividing and begins to enlarge and differentiate into a nitrogen fixing endosymbiotic organelle, which is called as bacteroids. So after the formation of the bacteroids in the cortex, now the nodule formation is going to be initiated. Thus, the nodule formation is mainly going to take place in the area of a cortex and the pericycle. The cells of a cortex and pericycle will now divide rapidly and will form a round structure which is called the nodule. This nodule is going to contain all the important elements that are required for the nitrogen fixation, like the nitrogenase enzyme. In this way, overall root nodule formation takes place. But behind this, there are many signaling factors and proteins that are supposed to be play a very important role in each and every step. The nodulation process requires the exchange of a series of signals between the plant and bacteria. The legumes secrete signals, usually flavonoids, which can passively diffuse across cell membrane, and the bacteria recognize these using a positively acting transcription factor, which is encoded by NOD genes. This interaction induces transcription of NOD ABC genes and other NOD genes. The NOD D protein binds to highly conserved bacterial promoters called NOD box and induces the expression of several genes. Many of these genes are involved in the biosynthesis and secretion of signaling molecules called NOD factors. The NOD factor induces root hair curling as well as it triggers plant division, which leads to the formation of root nodule. So these are the references which I have used. Thank you.